Hi, welcome everyone. So I'm Rob Howes, and you can see my short bio there. I've got background in oncology research, but also I've been instrumental in bringing the AAV technology into Horizon, and also latterly developing our academic network program where we enable labs with the technology. So I just wanted to really briefly cover what's Horizon's mission, and our mission really is to accelerate the discovery of novel medicines, so getting the right drugs to the right patients at the right time. And we feel that our genome editing technology can impact this at many stages, all the way through from the individual genetics through to developing the personalized medicines. Now, one of the real benefits of having this technology at this particular time is that we have a lot of information now from different genomics platforms where we've been able to analyze individual genetic variation and identify those which are disease-causing or disease-associated. And then we can use genome editing in either of two flavors, either nucleases or with AAV, to create isogenic cell lines. And we then use these for the development of either therapeutics or diagnostics, all leading to the development of personalized medicines. So the aim here is to get the right medicine tailored towards your particular disease based around the patient genetics and the understanding of those genetics in resistance and sensitivity. So what are our X-Man disease models? Well, these are isogenic cell line pairs where we take a parental cell line and we use our genome editing technologies to create variants of these. Now, these can be a wide variety of different genetic changes. The key point is that they're all made at the endogenous level, so that the control mechanisms for expressing these genes are the correct mechanisms within the cell. We end up with a pair of cell lines, and these can be used at many stages, from validating particular mutations all the way through to creating pathway reporter lines, through to creating complex disease models. So we think of these as surrogate patients or patients in a test tube, so because they respond exactly the same way as patients do. One of the key things with the technology is its flexibility. So what we do when we're thinking, especially with AAV, we think about the patient genetics as driving the type of genetic change that we want to make. So, for example, if you're interested in oncogenes, often these are single base pair mutations within exons in a heterozygous background. So we can exactly do that. We can replicate exactly the mutation and the genetic change that we see in the patient. We can also do gene knockouts. Now, this can be a variety of genetic changes, be a gene deletion, or it can just be a disruption of a gene. But we can also do a wider range of genetic modifications, some of the more complex ones, especially the type of modification which is found in cancer, such as large deletions, translocations, and amplifications. Now, what's really important with the AAV technology is that we've been able to target a range of parental cell lines, and we've done this for over 500 different projects, covering a wide range of tissue types. This is a technology that was historically used in mismatch repair deficient tissue types, but we've been able to increase the efficiency of the technology to develop it to target a larger range of more relevant tissue types. By having this technology, we've been able to produce a wide range of products and services based around the core technology, and you can see there's a range of them on this slide. Everything from creating the cell lines either off the shelf or a custom development, creating reporter kits, reference standards for the diagnostics arena, and then through to some of the drug discovery specific applications such as novel target identification and validation, research services, and even into the bioproduction arena. I mentioned before that we've targeted a range of cell lines in a range of projects, and here's actually it's a fairly old slide of a range of recent projects that we've completed, and you can see this really demonstrates the broad applicability of the technology. So on the left you can see a range of cell lines. Now, historically AAV was used mainly in colon cell lines, but you can see we've targeted a wide variety of other tissue types. And in the right hand column you can see the range of genetic changes that we've been able to make. So you can see that we've got everything from multiple mutations within the KRAS locus at 2 particular codons. all the way through to knockouts of specific genes, but also more subtle genetic changes such as knockouts of microRNAs. This is an example of all of the different types of cell line projects that we've recently completed. And as you can see from this slide, we've recently reduced our cell line pricing after some market analysis, where we've taken the cell line price to below $1,000. This price doesn't include shipping, but it does allow you to access the cell line for 3 years. We also have a range of other discounts available, depending on whether you are able to refer us to someone else or whether the cell line arises from a collaboration. There's also a small fee if you want to restock with a low passage number vial of the same cell line. One thing I want to cover next is the use of these in creation of endogenous pathway reporters. So this is a novel application of bringing together two technologies to really reveal a new type of assay format. So one of these is the X-Man Nolook reporter cell lines. So this is Nolook is a technology from Promigo, which is a new luciferase which is super bright. So it's very bright compared to the current luciferases that are on the market. Now the great thing about this is that because it's really bright, you're able to detect relatively small changes, and these small changes are the types of change in levels that you would get from an endogenous reporter. Now previously we could have engineered in standard either firefly or vanilla luciferase, but they were not really bright enough for us to be able to detect the endogenous changes. Because this luciferase is much brighter, then we're able to use this to create these endogenous reporters. The other technology that we've brought in as well is the Halotag system, again from Promigo. So Halotag is a small bacterial protein which has a covalent ligand. Now this ligand has been functionalized in different ways, either with a range of fluorophores or attached to a solid surface or even to reactive groups. This technology is really useful because it allows us to create endogenous protein fusions, so either N or C terminal fusions, that allows you to really investigate the endogenous levels of protein within your cell. The real beauty, though, of the system, because of course you can do that with an antibody, for example, but the real beauty of this system is that you can take the same cells and just by switching the ligand allows you to do different types of experiments. So, for example, if you use a fluorescent ligand, you can look at protein localization, but if you then use this a ligand which is attached to a surface, you can do a high affinity pull down. We've also reduced our pricing on the reporter systems, so both the luciferase and the Halotag versions, and you can see that we've recently taken this across so that the prices come to below almost to 10 cents per well for some of these reagents. Some examples of the cell lines that we use are the HIF1 alpha reporters, which allow you to detect the activation of HIF1 alpha in response to low oxygen conditions. And if you're looking for a kit that we don't have, we do offer a custom cell line service under several models and which should suit your particular approach. The next slide is some of the work that we've got in-house around our discovery research services. So this is a specialized group within Horizon that has access to all of the isogenic models that we have available. So access to all of the 500 plus isogenic cell line pairs. And these groups can be accessed on a project-by-project basis to do a range of projects, all the way from profiling target drugs under tumor microenvironment conditions through identification of novel targets through either SI or shRNAs. They have access to the endogenous pathway reporters, so if you want to look at the effect of a particular drug or knockdown of a particular gene on a pathway, this group can do that work for you. They can also then do in vitro and in vivo imaging and growth of these isogenic cell line pairs. And that's really one of the beauty of the isogenic cell lines, is that their ability to translate from in vitro to in vivo using the same cell lines. And that's of course really important because it's one less variable when you're trying to translate from the in vitro to the in vivo arena. So on the target identification and validation area, which I just want to cover in this, there's a range of work that we actually do. So we can use our isogenic models to create targeted gene knock-in or knockout. So we either can knock out the gene to see whether, if when we remove its activity, does that give you a particular phenotype that you're interested in, but we can also do more subtle changes. So for example, if you've identified a particular target which has some catalytic activity or has a disease-associated mutation, we can use our gene editing technology to introduce those mutations into your target cell line. And you can see that that's exactly what you see when you look at our online catalog. There's a range of these off-the-shelf cell lines that you can have access to. We also have within our contract research group, or even as an alliance, where we can profile your drug candidates either to identify novel targets but also to validate these targets. So for example, we can use an siRNA library to identify a novel target, but we can then use our genome editing technology to provide further levels of biological validation on that particular target. And we think this is really where we have a unique ability with our gene editing technologies to provide really high levels of biological validation before you would then go into maybe some chemical proof of concept trying to identify small molecule or biological inhibitors of the novel target. We've also got a consortium called Synthetics, which is based around synthetic lethality screening, which is some internally funded research, but also is part of a consortium where we do wide screens against the cancer genome to identify novel targets which are synthetically lethal in combination with particular cancer-associated genotypes. And this is something where we are looking for another partner within this, but really the aim of this consortium is to identify novel targets associated with particular disease-associated mutations, especially difficult to drug genotypes such as oncogenes and tumor suppressors. So the next section is the work we do with our academic collaborators. So we have a range of academic collaborators, over 100, and you can see there's a sample of them here on this particular slide, based essentially all over the world, and especially quite a few over in the Far East. Now we work with these groups across a range of different areas, and I'll go through each of these briefly. One of the ways we mainly work, though, is through our Centers of Excellence program, which I'll come on to later, so I can kind of skip over that. The other ways we tend to work, especially within the EU, is through some of the European-funded grants. Now, we're actually part of 4 of these, 2 of which are based on cancer, so both Collaborators, which is based around colon cancer, and Predict on renal cancer. Then we have some work around stem cells, part of the 4D sulfate. And more recently, we've just been awarded another EU grant based around cardiovascular disease. So here's one example in the Collaborators consortium, where we have a range of collaborators, so both within the UK and the rest of the EU, where we're trying to understand resistance mechanisms within colon cancer. Now, we've had a real impact using the isogenic cell lines, where we've shown some work that in patients that are taking drugs targeted against EGFR, if they have a secondary mutation in the KRAS gene, if they have a mutation in codon 12, they are resistant to the drugs. However, if they have a mutation in codon 13, they actually have sensitivity to these drugs, which is different to the data that was seen previously. And that's really impacting because these patients currently don't receive the drug. So this is now being taken into a prospective clinical trial purely based around the EGFR and KRAS G13 mutation status. Similarly, we've done some work in collaboration with one of our SAB members, Eric Hendrickson, where he's shown that modulation of the DNA repair pathways can increase the rates of relative gene targeting. So you can see that suppression of non-homologous end joining can really increase the rates of gene targeting from the relatively standard 1 to 3% range up to 10%. And that obviously has a massive effect on the ability for many labs to incorporate this into their workflow to create the isogenic cell lines. Because of course we understand that making the cell line is an end product for us, but it's just the beginning of the work for you guys. I mentioned briefly the 4D sulfate consortium, and this is looking at looking at proteins involved in the epigenetic polycomb repressor complex, but also the NERD complex. Our role in this is to create some endogenously tagged ES cells and iPS cells. And this is where this is going to be the first endogenously tagged stem cell cell lines, which we can then use to really understand, in this particular consortium, the role of these epigenetic regulators in disease development. I mentioned briefly some work we're doing with the endogenous reporters, and this is a collaboration we have with NCATS over in the US to really validate the endogenous reporters. We've done our own validation in-house, but we are working with groups over in the US to really see, can this be translated to higher density formats, so for example 384 and 1536 plate formats. And you can see we're testing a range of different endogenous protein and promoter readouts for a range of interesting genes, and we're hoping that the data for this will come out later this year. We've also been doing some work with the Sanger Institute here in Cambridge to use our isogenic cell lines. Now, they've been doing a larger project, which is on their website currently, the results of this, where they've targeted a range of non-isogenic cell lines with particular targeted drugs, trying to look for any correlation between mutation status and drug sensitivity or resistance. And they did this and showed a new use for PARP inhibitors. They're now taking our isogenic cell lines and doing exactly the same. So in a more reductionist methodology, trying to see is there a correlation between drug sensitivity and resistance and the isogenic and particular mutations. Now, I mentioned before some of the work around synthetic lethality screening, and this is something where we've been able to use our isogenic cell lines as a novel tool in synthetic lethality screening. So we can take difficult to drug genotypes, so for example loss of a tumor suppressor or an oncogenic mutation, create the isogenic cell line pair, and then screen against an siRNA library. So what we're looking at is when you knock down another gene, does that give you a selective effect for a particular genotype? So here's an example where we took a tumor suppressor P10 and we did a cell-based screen using an siRNA library to look for targets that selectively kill cells with a mutant P10 compared to cells with a wild-type P10. Now, P10 is commonly mutated in cancers, so of course you're then looking for a synthetic lethal interaction for a target which will selectively kill the P10 mutant cancer cells and not the wild-type tissue in the same patient. And you can see here's an example where NLK shows a synthetic lethal interaction with P10 deficient tumor cells. And that's the nice thing about this, is that it holds up across a range of cell line backgrounds. It's not just a cell line specific effect. A couple of slides now on the Center of Excellence program. So this is a program which is really aimed, it's got two main aims. One is to enable academic groups to use the AAV technology. So we know that AAV is the kind of it's a less well-used technology compared to genome editing technology compared to some of the nuclease technologies. We also want to expand the use of AAV outside of oncology. So most of the people within Horizon have an oncology background, and that's where we've really used the technology in the past. But we know that it can be applied across other disease areas, so we're trying to really reach out to other core groups to use this technology to create novel disease models outside of oncology. So the program's been running for just over 2 years. We've got over 60 different labs that are using the technology. Over 150 projects are ongoing, and we have the first sets of cell lines that are coming back through that have entered the Horizon Bank. So one of the key features of this program is that every cell line which gets made after publication comes back into Horizon, where we can then do our own cell line validation and have those cell lines available for people to access. You can see the location of the groups across the whole of the main areas in the world, as you would expect, with the orange blobs being the kind of core centers. So these are areas where not only are they particular labs who are using the technology, but quite often these are core facilities. So this might be a core lab which is then taking in projects from many different groups within that particular institution, generating the isogenic cell lines, and then passing the cell lines out to those researchers for their particular studies. You can see that we've started to expand across different disease areas. Again, most of them, 2/3 of them, are within oncology, but you can see we've started to expand this into other disease areas. And here's just a couple of examples that I wanted to flag. So cardiovascular disease. This is a disease which is probably at the stage where cancer was 15, 20 years ago, where there are some disease-associated loci, but we don't know whether those loci are actually causative or just associated. So we can use our gene editing technology to create isogenic cell line pairs to really understand, okay, are those disease-associated mutations actually causative or just associated? Similarly, in neuroscience, we're trying to use our isogenic cell line models to create better disease models for some of the triplet repeat diseases. So, for example, in Huntington's, because the Huntington gene locus is so big, it's almost impossible to create a cDNA overexpression model. So the way that people currently do this is that they just express the first exon, and it's the first exon which has the expanded triplet repeat within it. So within Huntington's and other triplet repeat diseases, in normal wild-type patients, you tend to have a low number of copies of this repeat. And as you get more copies, you get a more severe form of the disease, which is more penetrative. Now, what we're trying to do with our isogenic models is instead of overexpressing just the first exon under a non-endogenous promoter, we can target the endogenous gene. So we can create an allelic series. So we can take a cell line which is wild type, in this case for Huntington's, and we can then create increased numbers of the triplet repeats at the endogenous locus. And this is really important, of course, because that allows us to look at stepwise at what point do the increased number of repeats lead to the particular stages of the disease. We're also then working in different disease areas, so stem cells, can we create Linda reporters or patient disease models, but also other types of disease areas, such as autoimmune diseases. So that's a brief overview of the academic program. If you want any more information about the program and how you can access it, just drop us a line or maybe contact us after the webinar, and we can send you some more information. One of the great things about this particular program is it's had a real impact. So this was an article that came out last month in Nature Biotech, looking at the most active pharma in their collaborations with academia. You can see there's a really nice quote where we closed 5 academic deals last year, which is actually more than some really quite high-profile pharma companies such as Bayer and Boehringer Ingelheim. Now, funnily enough, the 5 academic deals that we closed were actually the ones that we talked about. There were probably another 5 or 10 which we didn't actually publicize. So if you actually included all of the academic deals that we did last year, we'd probably be up in the range of GSK or Pfizer. Okay, so that's the end of my section. I just want to now pass over to Ian Pryor, who's going to give us some background. So a short presentation on the work that he's been doing with the isogenic cell lines. So Ian's been working with us for just over a year now. He's based over at the University of Liverpool, and his lab is really focused on the different isoforms of Wrass and trying to use proteomics to understand how the different Wrass isoforms signal, and especially the mutated forms in cancer. So I'll hand over to Ian, and he can give you the short presentation.
Okay, thanks, Rob. So I'm using isogenic cells for looking at Wrass signaling, and as Rob said, I've been involved with Horizon for just over a year now, and my background actually is I've been a group leader for about 10 years, and I use a variety of approaches to look at Wrass signaling and compartmentalization of signaling pathways. And one of the key issues within signaling is that, most of the techniques that we use to study signaling involve overexpression of our proteins of interest, and this obviously has perturbing effects even if we overexpress the wild-type proteins. And so as soon as I became aware of what Horizon were doing, this was obviously a very attractive model system to be able to apply to questions around cell signaling, because we no longer have these overexpression artifacts that we've been used to in the past. And so, as a consequence, we've been able to set up a Centre of Excellence in Liverpool, and through collaborations with Horizon, we've been able to start to develop our own cell lines of interest. And so I'm going to talk to you now about some work that we've been doing with some of these cell lines and the types of approaches that we're using to investigate the biology that we're interested in. And so I'm sure you're familiar with the Wrasses pathway, but just in case, Wrasse sits downstream of cell surface receptors, such as the EGF receptor. It cycles between inactive GDP-bound conformations and active GTP-bound conformations, and it actually activates a whole variety of effectors, but principal among them are the kinases, such as the RAF MAP kinase pathway and the PI3 kinase AKT pathway. And these have regulatory effects on a whole variety of cell phenotypes, such as proliferation, differentiation. And in the cases of cancer, what happens is that RAS becomes effectively permanently GTP bound. It's unable to switch itself off very efficiently, and so you end up with hyperactivation of RAS pathway, RAS effector pathways, and so this then promotes things like proliferation and differentiation. And so this is in the context of a cancer. If we were overexpressing RAS, what can happen is that instead of promoting proliferation, you can end up with things like senescence, and so this is potentially an artifactual situation caused by overexpression. So again, this is one of the reasons why we want to be able to look at the endogenous signaling rather than the perturbed overexpressed signaling. So my own interest in RAS is spurred because all cells express 3 isoforms, and so there's HRAS, KRAS, and NRAS present in every cell, and these are almost identical proteins. And the region of identity actually corresponds to the areas involved in the function of RAS in terms of the effector binding and also the nucleotide binding. So these are the bits that are responsible for the on-off of RAS and also for the interactions that with downstream effectors. And so in vitro, these proteins are identical. However, we know that there are biological differences between these proteins, and one of the key differences between them is their contribution to cancer. And so this is just a survey of the Sanger dataset where they've collated together all of the mutation frequencies of the different isoforms across a whole variety of common cancers. And what you can see here is that in black, all of these black bars here represent KRAS mutations. So if I just show you large intestine here, so colon cancer. So approximately 35% of colon cancers have a RAS mutation present, and the majority of these mutations are in KRAS. And this is actually a distinctive feature of RAS proteins, that different cancers appear to prefer a different isoform to be detected, and the reasons for this are currently unclear and something that a variety of groups are actually interested in understanding. But as a general take-home point, KRAS is the one that in many cancers is the key RAS contributor if a RAS mutation is present. So KRAS, for whatever reason, you could view as being particularly oncogenic of the 3 isoforms. So in my lab we have some questions around KRAS, and one of the further interesting features of KRAS is that it's actually alternatively spliced, and so you get 2 variants of KRAS that can be expressed. So these are called KRAS 4A and 4B, and that's because the exons that code for the difference are actually the 4th exon of the protein. So this would actually be the 4A or the 4B exon, depending on what's spliced in. And this region of the protein actually is responsible for the membrane binding and post-translational modifications of this region of the protein, and this results in differential targeting of these proteins to different subcellular compartments. But whilst KRAS itself is a fairly well understood protein, these specific isoforms and the relative contributions of each of these isoforms to the function of KRAS is a much less clear area. And so this is something that we've initiated a project to look at with support actually from Horizon in terms of helping to make these cell lines. So just to give you a little bit more information on KRAS 4A and 4B, what we already know is that KRAS 4B is ubiquitous. It's found in every cell, and it's the most abundant isoform in most tissues. However, work from Charles Patek's group, that Sarah Plowman actually performed, showed that 4A is also present in most tissues, but actually it's not ubiquitous. So there's a few tissues where there's actually relatively little, if not no, expression. So for example, heart is classically one of the tissues where there's supposed to be no KRAS 4A expression. But generally, especially in things like large intestine and stomach and some of the key areas where KRAS is known to be mutated, in cancer there's a lot of KRAS 4A present. There's also been some limited study of the function of these isoforms, and so this is based on overexpression, so this is kind of a classical way of looking at isoform-specific RAS function. So although the expression isn't equivalent here, and this is a potential issue, what you can see is that when different isoforms have been transfected into these cells, and then a kinase assay has been done for RAF activity, you can see that the two KRAS isoforms are more potent than the other isoforms activating RAF-MAP kinase. So this suggests that the different isoforms might be differentially coupled to the RAF effector pathway. So this represented the state of the art about 10 years ago. What we'd like to do now is to be able to ask that question in more detail. You know, what is the real difference between KRAS 4A and 4B? And so just a few more points really on why we're asking this question. We already know that KRAS 4B is essential for mouse development. Mice die if KRAS 4B isn't present. However, KRAS 4A mice that express KRAS that have loss of KRAS 4A are still viable. It's also known, there's some preliminary data really that suggested that in colorectal cancer cells where you get oncogenic KRAS mutations, that you may get changes in the ratio of KRAS 4A to KRAS 4B, and with a favoring of KRAS 4B expression. And also, there's a potential role for KRAS 4A as a tumor suppressor, where there was a loss of KRAS 4A in mice where these have been engineered to lose 4A. There was an increased number of tumors and the size of tumors that were induced in this model compared to one where both isoforms are present. So it suggests that there's an interrelationship between the isoforms, but potentially that KRAS 4B is the key isoform because loss of this one especially is a lethal phenotype. So our questions are, what are the functional differences between KRAS 4A and 4B? Can we study that in an endogenous context? And also, I mean, this is a more speculative question really, but you know, what is it that makes KRAS 4B more oncogenic than KRAS 4A? And the strategy that we've got with the tools that are available today are, what we'd like to do is to be able to measure endogenous isoform-specific signaling and to measure the network response to these pathways, to these isoforms. And so the strategy that we're taking is to engineer isogenic cells that express either 4A or 4B. We're using quantitative phosphoproteomics to look at network response, and we're also using phenotypic assays as well to look for differences in classical RAS function associated with these different isoforms. So just to give you an overview of the kind of ideas that we have for targeting and generating this and the technology, what we start off with is your target gene. In the case of RAS, we've got these 4 exons. We generate a vector where this vector includes homology arms that target the target DNA of interest, and within that you can engineer a change. So in this case we could, for example, include, instead of the 4B exon, we swap it for the 4A exon, so that when you get recombination, the 4B exon is swapped for the 4A exon, and you end up with a new cell line that contains the RAS gene as it was before, the KRAS gene as it was before, but now with a 4A exon in place of a 4B exon. And so then regardless of the splicing, you could expect to get 4A expressed. So that was one idea for how we might generate these types of cell line. Another idea, this was, we've actually got a variety of plans that we've talked through with Horizon. We started to make some of these cells now, is to replace the 4A exon in this case with a Neomycin resistance cassette. So now instead of splicing in 4A, you just splice in this Neomycin resistance cassette, and if it splices to 4B, that carries on as normal. So in this case, you can only ever get KRAS 4B expression. So obviously in the case of KRAS, there's 2 copies of this gene within the genome, so we would have to repeat this targeting for both alleles to be able to get full exchange of these exons, so that then we can only ever get 4B or 4A. So as Rob said before, this is a 3, 4, 5 month process depending on the cell type, and so you're looking at approximately 9 to 12 months in order to generate one of these cell types. Okay, so and the types of assay that we want to apply these cells to, and that we've also applied other X-Man cells to that we've already had from Horizon, are to use, for example, Cellular-based quantitative proteomics. And so this is a quantitative proteomic method that allows you to have different experimental conditions. So this could be plus and minus stimulation, or it could be, in our case, different cell line variants. These are labeled with isotopic variants of arginine and lysine. They're then mixed together. They go through a fractionation process, and then at the end, these samples are run on the mass spec. And The benefit of this approach is that all of the processing that occurs here with the fractionation and enrichment for proteins of interest, for example in our case the phosphoproteomic network, this can take a few weeks to perform, and there's a lot of steps where you get experimental variability or technical variability between runs. However, because these samples are mixed at the very beginning, all of that is then equalized, and so the ratios that we see at the end represent true differences that were present within this cell, these cells at the beginning. So for example, if this was RAF, phospho-RAF we were looking at here, this would suggest that in condition B, phospho-RAF was particularly active compared to condition A and condition C. So this type of approach, because of the scale of it, gives you a couple of thousand outputs rather than just one or two, which is the traditional Western blot approach, which allows you to get more of a view of the network, to sample the network that's downstream of your protein, or your pathway of interest or your perturbation of interest. So this is a bit more of a detailed overview of the type of methodology, the pipeline that we've got. So we're taking our cell line. So for example, in this case, this could be a KRAS cell line. It could be anything really. I mean, we've also got different variants of RAS as well that we're looking at. And you can put these through these types of phospho and proteomic approaches. So in this case, this is the phosphoproteomic arm where we're enriching for phosphopeptides. And in this case, this is the proteome arm where we're just going to cut this gel into pieces and then run this at the end and get details on protein abundance changes, and that these two points together help us to understand the network response. Some of the changes in phosphoproteome are a consequence of changes in proteome. There's more protein present, so there's also more phospho present, and in other cases they're independent, so there's specific activation of a phosphosite that we can discriminate based on these two arms of our protocol. And as you can see, this is quite a long drawn-out protocol, so we don't do too many of these, and there's also a potential here for variability that we want to reduce as much as possible, and that's one of the benefits of SciLac, that it really gets rid of a lot of the variability. The other benefit is the isogenic cells in the beginning. So for example, if we were doing this in a transfection configuration, every single time you do a transfection, you get different transfection efficiencies, so that in itself could cause difference. And so because we have isogenic cells, that effect has all gone away. We really are comparing things that have been standardized to the level we need to be able to make meaningful conclusions at the end here. And this is just an example of the kind of data we're looking at, and really to highlight that point. So this is a cross-correlation analysis where we're comparing three different cell variants with each other. And we've got the proteome and the phosphoproteome. And really what the point here is, first of all, the experimental reproducibility because of the use of isogenics. And so if you look at these squares here, these represent the cross-correlation between biological replicates of a particular cell line, and red means good correlation. And so you can see that when you're comparing the biological reproducibility, this is very good. However, when we compare the cell lines with each other, you can see that cross-correlation decreases, and there's a particularly profound decrease in cell line C versus the others. And so what this is saying is that cell line C is very distinct in its responsiveness compared to cell lines A and B. But even the difference between B and A is about five-fold difference in cross-correlation. And so in terms of the phosphoproteome, we can again see a similar pattern. Again, A and B are more similar to each other than they are to C. And the only reason really that I'm happy to trust these differences is because of the strong biological reproducibility that we've seen. And I think that is a consequence of using the isogenics. And then also the fact that we're not overexpressing, and so we've been able to follow up particular differences that we see within this data, and we see that in a variety of the isogenics that we've got, that we get very good reproducibility, we get very good confirmation in independent cell lines, independent clones of the data that we're seeing within our initial screen. Okay, so that's it for me. I think there's a final slide here, and I'll just pass you back over to Rob.
That's great, thanks Ian. That's really good to see some of that data where we can actually see the use of the cell lines, because of course at Horizon we focus on generating the cell lines, but what's really important is how people then use those in their particular research.